Welcome to the Metabolic Classroom. I am Benjamin Bikman, a biomedical scientist and professor of cell biology. Today we're talking about microplastics. That is the focus of the lecture. Now, microplastics, of course, are these very, very small, microscopic, in fact, particles of plastic that are generally not only beyond your ability to see them, but also beyond your ability to avoid them. They're virtually impossible to avoid in today's environment. And that's a result of what we eat and drink and what we put on our skins. So whether it's because of microplastics in the oceans or in the food we eat or the water we drink or the ointments and lotions we're putting on our skin, we really can't avoid them. But interestingly, while they are practically unavoidable, people are generally totally unaware of them. So today, my goal is in this lecture to just dive into how these particles might affect human health, with a particular focus, of course, on metabolically relevant tissues and processes. So a heavy emphasis on the effects of microplastics and their chemicals, on fat cell biology, which is, of course, a cell type I focus on quite heavily as a scientist, and in some of the consequences of this, like insulin resistance. I also wanted to make a point to talk about the relevance of microplastics in blood vessel health and blood vessel physiology, because I actually think it's quite relevant, and there's some decent findings to support this. So, by way of wrapping up the introduction, we're going to explore the science behind all of these effects with a particular focus on some key chemicals within these plastics that get leached out of the microparticles themselves, namely BPA and phthalates. These are two of the major chemicals. All right, so let's get started. So first and foremost, let's just take a moment to actually define microplastics. So the definition here from a sort of chemical view is that microplastics are very small plastic particles that are less than 5 millimeters in size. So this is an exceptionally teeny little bead, and they come in two forms. Generally, they come as primary microplastics, like little microbeads, and these can be things deliberately put up that way, like made that way, like in cosmetics. And there are secondary microplastics, which are small fractions that have been sloughed off of a larger plastic item, like a bottle, a plastic bottle, or a plastic bag. Now, I'd mentioned earlier how ubiquitous they are, and microplastics are virtually everywhere, in oceans, soil, air, even in what we're drinking. Studies suggest that the average person could ingest or inhale even, these are small enough to be airborne, up to 5 grams of plastic per week. So that's about the size of a credit card. Now, lest you think, well, that's not that much. To consider that that's how much plastic is working its way into your body is I think a little more sobering, or I hope it is at least. Now, as I mentioned, with human exposure, our main routes of exposure do include ingestion, things that we are literally putting in our mouths, like food and water, but also possibly through the skin, where these things could be small enough at the nanoplastic size, not just microplastic, but even smaller, to actually penetrate skin and get into tissue and blood that way. All right, so that is what they are and where they come from. Why do they matter? Why is a metabolic scientist taking time to talk about this? Because they are relevant for metabolic health as they accumulate in tissues. Now, as microplastics have become more talked about, by now you've likely heard that microplastics also accumulate in the gonads. Now, the gonads is the general term for the key reproductive endocrine organs in humans. Males have testes, females have ovaries. Those are the gonads as they sort of commit to one path or the other based on the genetics of the baby. I wanted to briefly mention those. That's beyond the scope of the metabolic classroom. This is, after all, not the reproductive classroom. But I wanted to touch on this because it is something that's of such interest at the moment. So as much as the microplastics don't just pass through us, they do accumulate in tissues like fat tissue, but also, as I noted, the gonads. So let's just briefly review that. So one particular study on females, on female rats, found that microplastics actually could disrupt ovary function. In particular, they found that there were alterations in the physical structure of the ovaries, as well as some of the proteins within the ovaries that are considered what's called cytoskeletal. So this provides an actual physical structure, like a scaffolding within the tissue. The net effect of this was that egg development oocyte, which is the technical term for these eggs, was compromised, and the actual number of eggs left in the ovary after exposure to the microplastics was significantly reduced. That, of course, is catastrophic, given that there are a finite number of oocytes within the ovaries. And if you have the microplastics coming in and decimating them, that absolutely has consequences for fertility. Also has consequences, mind you, for the onset of menopause, which is generally a function of running out of eggs. Okay, now what about in the testes with testicular toxicity and male fertility? In male experimental models, microplastics affected, once again, testicular structure and function, including apoptosis, or the programmed death of cells like Leydig cells or Sertoli cells. Those are some of the cellular components, indeed the main cellular components of the testes, but also resulting in the reduction in spermatogenesis. So the actual production of sperm. This is the germ or the reproductive cells, of course, in the males, the parallel to what is the oocytes or eggs in the ovaries. So good reason to focus on gonads, even though this is not the reproductive classroom, this is the metabolic classroom. I felt compelled to mention that as just an interesting point of science and as a person who is an enormous advocate of people having babies. All right, let's move into some more specifics of the plastics, which allows us to transition then into talking more about fat cells and the compromised function. So I mentioned earlier that a lot of the problem with the microplastics is really twofold. It is the physical presence of the plastic itself, and then it is the chemicals that are coming off of the microplastics or getting leached out, particularly BPA and phthalates. So many of the microplastics contain endocrine disrupting chemicals like BPA and phthalates, as I've mentioned now multiple times. There are more, but those are the big ones. These chemicals disrupt the endocrine system, affecting everything, as I noted, from reproductive health to metabolism, because metabolism is so affected by hormones. That's one of my main areas of research and areas of teaching, is just to help people understand that metabolism is far more complicated than just calories in, calories out, that there's an entire endocrine component to it. In other words, hormones that tell cells what to do with the energy they have access to, most especially fat cells. So these endocrine disruptors, these molecules that are coming from the plastic, can mimic or block natural hormones. In some instances, as I'll outline some of the reasons here, are referred to as obesogens, so chemicals that actually promote obesity, once again suggesting that there's more to the story than just calories. All right, now let's transition and talk about how BPA and phthalates in particular disrupt fat cell function. So as I mentioned a moment ago, there's a hormone mimicking aspect, and BPAs mimic estrogens by binding, and then they can bind estrogen receptors and disrupt normal hormone signaling. Now you'll notice my language, I'm saying estrogens. There's no singular hormone called estrogen. It's a small family of these prototypical female sex hormones. Males have them too, of course, and they serve a function in men as well, just not quite as important as a function that they serve in females. Phthalates, on the other hand, are more specific to disrupting testosterone and related hormones, testosterone being the poster child of the androgens, the prototypical male hormones, although once again, females have androgens as well, and they are critical. But the net effect of this is a change on fat cell metabolism and overall metabolic regulation, because these sex hormones do have an effect. Testosterone signaling general favorably changes in burning fat. Estrogens also actually facilitate this signaling to break down fat and also store more fat. So, as a brief tangent, women have a much higher turnover of fat in the fat cell. That as much as a woman is in fact burning or breaking down fat much more readily than her male counterpart is, she's also storing more at the same time. So there's a greater turnover. All right. Now that's the sex hormones, which is still kind of making you think about the gonads, which is fine. But I really want to move us firmly into the fat cell and highlight how these chemicals are involved in adipose or adipocyte. Adipocyte is the technical term for a fat cell. All right, so both BPA and phthalates activate a key transcription factor in fat cells called PPAR gamma. Now, when you hear the term a transcription factor, you can think about like a fuse, a central kind of electrical box, where if that fuse is off, then any of the light switches, like let's say I go out to my garage and I turn off the fuse for the kitchen lighting. Now, if someone's inside the kitchen and they're flipping the light switch on or off, it's not doing anything because I've turned the main fuse off. That's somewhat analogous to what PPAR gamma is doing, where it is a key transcription factor. It basically is then going to affect the function of anything downstream, any other genes that are going to be involved in telling a fat cell to grow and or multiply. So with PPAR gamma, it's the fuse, and now it's been turned on. So this has given other genes, like the light switches in my kitchen, the ability to also now be turned on. It's empowered subsequent steps. So, as I mentioned, both BPA and phthalates both activate. They turn on that master regulator in the fat cell called PPAR gamma. This factor is also connected to another one called the CAT enhancing binding protein alpha, or just abbreviated as CEBP alpha. This is once again a critical adipogenic gene. Adipogenic or adipogenesis is the signals that are promoting fat cell development. In other words, taking stem cells and telling the earlier young cell, you young cell, I need you to become a fat cell. So it stimulates this development or maturation into a fat cell. And also, of course, in that process, telling fat cells to store more fat. All right. Hopefully that makes some sense. But when these pathways are activated by BPA or phthalates, it signals the fat cells to store more fat, to multiply, resulting in an overall greater capacity of the body to store fat. In other words, the body has gotten fatter through both more number of fat cells and larger fat cells. One of the studies that I'm pulling this from was published in Environmental Health Perspectives, and they found that BPA exposure significantly increased some of the metabolic complications in humans that we'll get to more, like insulin resistance, which of course is heavily influenced by fat cell size in particular. Okay, so that's some of the signaling. There are also, interestingly, beyond that, changes in some of the hormones that fat cells can make. And very, very briefly, what has been shown is that BPA and phthalates have a very unfortunate change in fat cell endocrinology. I've talked before about two critical hormones that come from fat cells: Adiponectin and leptin. You're very familiar with leptin. People know about it in the context of a satiety signal, although it does much more than that. Adiponectin is one you're a little less familiar with. It's not talked about as much, but it's a powerful signal from a fat cell that promotes insulin sensitivity throughout the body. What happens when the fat cells are exposed to the microplastics and its associated chemicals is that adiponectin production plummets. Interestingly, leptin production goes up. That is, in fact, a pathological change. That's a change that suggests an unhealthy shift in the biology of the fat cell. Because adiponectin has such benefits, as it starts to go down, it's a sign of fat cell hypertrophy, which we're going to talk more about, of course. As leptin goes up, it's a sign that overall fat mass is going up, because as fat mass goes, so too does leptin. But there's this direct stimulation of these microplastics and chemicals of stimulating leptin. As another little tangent, I think that may be the second one. I've talked about this before. One of leptin's many effects is to stimulate the hypothalamic pituitary gonad axis. In other words, it tells the brain to tell the gonads that it's time to initiate puberty. One of the reasons why we may be seeing such an epidemic of precocious puberty, in other words, kids going through puberty earlier, is that these microplastics may be stimulating the fat cells to create more leptin. And as there's more and more leptin, it tells the brain to initiate puberty. In this case, perhaps a little too early. So that has direct effects throughout the body. As you are having the microplastics and the associated chemicals changing the hormones being produced by the fat cells, you're messing with some pretty powerful signals that go beyond just metabolism. Okay, now we're not done with the fat cell yet. So one other thing to talk about here, well, among a handful still to come, studies have found that microplastics, particularly through these chemicals, the BP and the phthalates, do accumulate in fat tissue. And when the fat cell gets invaded with these chemicals and the plastics themselves, it activates inflammation. So the fat cell has basically consumed something that it doesn't know what to do with. And its emergency response is to start belching out pro-inflammatory proteins, which are called cytokines. Things like TNF alpha or the interleukins. You can look these up for more details. They are canonical pro-inflammatory signals. These will then circulate through the body, promoting systemic inflammation and, of course, insulin resistance, insofar as inflammation is one of the cardinal causes of insulin resistance. But this is relevant because we know that there are that in humans we can have something called chronic subclinical inflammation. In other words, a patient comes in and they get a blood test and you see something like their C-reactive protein, a cytokine, levels are elevated. And yet not to the point where they have an obvious infection or they have mono or something like that, but also not at a healthy level. They're elevated and demonstrably so, even though the body is not fighting an infection. It's almost as if the body is fighting an infection that's not there. That's because the fat cells just don't know what to do. They're panicking and they're releasing all these pro-inflammatory cytokines as they have something in them that they can't get rid of. In fact, I'm going to come back to that idea more in a moment when we talk about macrophages. Okay, now still about the fat cell. A key process that I've alluded to today already and multiple times in previous Metabolic Classroom lectures is this process called adipocyte hypertrophy, the enlargement of individual fat cells. Adipocyte hypertrophy is more pathogenic than simply adding new fat cells or hyperplasia. When adipocytes grow in size, they become highly metabolically dysfunctional. They become very insulin resistant to restrict further growth. And I've talked about this before, so if you need more background, please go watch some previous episodes. And they also become very pro-inflammatory, separate entirely from the effects of the microplastics and the chemicals. When the fat cell is undergoing substantial hypertrophy, they start to suffocate, or at the cellular level, we use the word hypoxia. They become hypoxic. And one way to try to correct that hypoxia as they're getting pushed too far from blood vessels, from capillaries, and the life-giving oxygen that comes from them, they will secrete these pro-inflammatory cytokines in an effort to stimulate the growth of new blood vessels. So it's a way of the fat cell trying to correct its hypoxia. You gotta empathize with the poor fat cell. Again, it becomes insulin resistant to try to restrict further growth, and it becomes pro-inflammatory to try to correct the hypoxia, to give itself more blood flow. As a sort of important addition to that, when a fat cell becomes insulin resistant, that creates a unique scenario within the body where insulin is high, which it is during insulin resistance. Remember, there is no insulin resistance in the absence of elevated insulin. Those two things are just locked hand in hand. So insulin is high, but now the fat cell, normally if insulin's high, insulin is telling the fat cell to hold on to its fat and not be breaking it down. But when the fat cell becomes insulin resistant, it stops listening to that signal. Now, even in the midst of elevated insulin, it's breaking down its stored fat and releasing it as free fatty acids. Now you have this metabolic paradox where insulin is high and free fatty acids are high. This leads to the so-called problem of lipotoxicity. I don't love that term. What I prefer would just be saying ectopic fat storage. So now you're forcing the body to store fat in places where it doesn't want to be storing fat long term, like the pancreas, like the liver, like the muscle tissue. Now you'd say, well, why don't those tissues just burn the fat? Because insulin is high. If insulin is high, you can't burn that fat. You're forced to store it. All right, hopefully that makes some sense. I worry that I've gotten a little too complicated there, but that's just that's finishing the thought of why the hypertrophic fat cell, as it becomes insulin resistant, is so harmful to the rest of the body. All right. Now, there is some animal studies that have looked at this where you can just do a little more with animal tissue than you can human tissue. That's no surprise. But in response to BPA, there's one of the studies I've highlighted here in the list of references showed that fat cell size went up by about 30% in these animals just because of the BPA exposure. I mean, 30% is a pretty not only statistically significant, which enabled the scientists to report it in the first place, but I would submit that it's also meaningful. Not all statistically significant findings are actually meaningful, but a 30% increase in the size of a fat cell is meaningful. And they found that this was driven by activation of PPAR gamma and CEBP alpha, which again are telling the fat cells to take in and store more fat. So the fat cell becomes very swollen, which means it becomes insulin resistant, which then in turn leads to the fat storing more fat in tissues throughout the body. Okay, let's just go a little. I've talked enough about the fat cells, how they become insulin resistant to restrict growth, and they become pro-inflammatory to correct the reduced blood flow. So that's enough on that. So hopefully by now you have a pretty good feel for how microplastics and BPA and phthalates, the chemicals of the microplastics, are promoting the proliferation or hyperplasia and the growth of fat cells. Those two are a wicked combination, making a body be much fatter than it was before. In addition to, we have the changes in fat cell hormone production, like adiponectin going down, leptin going up. This is actually a surrogate marker that I've used in my lab. We just published a paper looking at this. Others have too. I didn't make it up. I adopted this protocol from the strategy from others. Sometimes when you can't measure, like in humans, it's difficult to get a measurement of fat cell size. You have to do a fat biopsy, then you've got to process that through a process called immunohistochemistry and get a picture on the microscope of the fat cell. We do that. We're doing that more and more now with my collaborator, Dr. Paul Reynolds, but it's not easy to do. What is easy to do is get a blood sample and measure adiponectin and leptin. As the adiponectin to leptin ratio, adiponectin in the numerator on top, leptin in the denominator, as I outlined earlier, adiponectin was going down, leptin was going up. That general shift, if the adiponectin to leptin ratio is going down, that suggests adipocyte hypertrophy, that the fat cells are getting bigger, because that is the exact change in hormone production that you see as fat cells are growing. So as a fat cell gets bigger, its adiponectin production drops, its leptin production goes up. So that's a surrogate marker of adipocyte size. And indeed, that's what we see. So hopefully by now you're pretty comfortable. What we've talked about is what the microplastics are and their related chemicals. where they come from, and now the specific effects, especially on fat cells, where when these chemicals get in, they are directly activating these master gene transcription factors. They're turning on the fuse box, promoting, ultimately signaling to the fat cell, you need to take in more energy and get big and multiply. That's a wicked combination that goes way beyond just what your calorie count is in or out. All right, now I alluded to this idea of the blood vessel wall as well. I just wanted to end with that thought because it's so fascinating. So while much of what we've talked about is focused on the fat cell, and the fat cell itself has an effect on blood vessel health because of insulin resistance and because of inflammation, there is this direct idea of the microplastics invading the vessel wall. Now, by way of background, heart disease is the leading cause of death in much of the world. Most of the, I would say industrialized, but even developing countries, heart disease is the number one killer. Part of the pathology of heart disease is the development of the atherosclerotic plaque. You've heard of atherosclerosis, you've heard of plaques. These are these growths within the wall of the blood vessels, particularly the blood vessels of the heart, the coronary arteries, that the plaque is expanding, and then ultimately blocking the blood vessel. Then you have an infarction. If that happens anywhere else, you'd have a stroke. So that's when you just restrict the blood flow to the tissue. So as much as that is the known problem, what is not known is the actual etiology or the precise steps that cause the plaque to form in the first place. We have a lot of theories. The one you've heard before is that cholesterol gets into the blood vessel wall and that creates a plaque. That is absolutely overly simplistic and wrong. There is evidence to suggest that it's because of just infections, that you have bacteria that infect the blood vessel, and that results in this immune response and a subsequent accumulation of immune cells like macrophages, and then the plaque is growing. Another idea is that you have microplastics and nanoplastics that are invading. So let me just touch on that a little bit. There was a notable study published in the prestigious New England Journal of Medicine that actually analyzed the composition of atherosclerotic plaques from patients that were undergoing procedures to remove plaques. Now, what did they find? Some of the key findings: the study detected these polyethylene microplastics in almost 60% of all of the plaques that they sampled. So 60% of the plaques had these specific microplastics, the polyethylene version, and then polyvinyl chloride, which is another microplastic variant, was found in another 12%. So we're getting up to almost 3/4 of all of the patients that had carotid atherosclerotic plaques sampled had confirmed presence of these micro and nanoplastics. And then when they did some advanced microscopy, to really zoom in, kind of like what I was talking about with the fat cell earlier, how it's a little complicated to actually get a picture of the fat cell. When they did that same procedure with these atherosclerotic plaques, they found the presence of these like teeny, like 1 micron, like a micrometer in size, these jagged little plastics. And in particular, what is so important is that a known component of atherosclerotic plaques is the presence of these type of macrophages called foam cells. So a foam cell is basically this fatty macrophage. But in this case, what was happening is that the jagged little microplastic or nanoplastic would embed itself in the wall of the blood vessel. That is damage. One of the cells that responds to damage is the macrophage, trying to act like a hero here. And so the macrophage will come to the wall of the blood vessel, and it has an ability to move through the blood vessel wall. It can flatten itself out and then kind of slurp down like through a drain. And it can get to these jagged little microplastics and start eating them or engulfing them or phagocytosing them. A phagocyte is a term for PHAGOCYTE. Phagocytosis is the process of a cell eating another thing. And so the macrophage is coming in and engulfing these jagged, harmful micro and nanoplastics. But in the process, as it's eating more and more of them, it becomes a foam cell. And the foam cell is broadly acknowledged to be the primary cellular component of the core or the center of the atherosclerotic plaque. The problem also is that when one macrophage starts to eat stuff, it starts to release pro-inflammatory cytokines, which in this case act like a trail of breadcrumbs for other macrophages to follow. It's basically one macrophage's way of calling out for its friends to come and help it, saying, hey, there's a lot of microplastics here. They're jagged. They're damaging the endothelium. I need you to come help me. Then you have more and more macrophages eating more and more jagged little sharp microplastics, and you get more and more foam cells. And now you're left with the core of the atherosclerotic plaque. So again, direct evidence of the presence of these plastic particles in almost 3/4 of all of the plaques that were sampled. Okay, now let's start to kind of wrap it all up. When we start to connect the dots, we do see a clear pathway that microplastics, the exposure to the plastics themselves and their chemicals, promotes hyperplasia and hypertrophy of fat cells, making them inflamed and insulin resistant. This systemic inflammation and insulin resistance originating from the fat cell, but also perhaps happening in blood vessels, as we noted, starts to increase the risk of diseases like type 2 diabetes and heart disease and other metabolic problems. Because the microplastics are so prevalent, it really is difficult to avoid. But I do think if I can leave you with any sort of strategies, there are, of course, some things that you can do, including being careful with the types of detergents and soaps you use to see that they're phthalate and BPA free, including the types of plastics you're using, what you're drinking your water from. It's wonderful. You know, one of the more bizarre trends for me as a father is to see how obsessed kids are, and a college professor. When I was a student, no one was walking around with a bottle of water. We just drank from the fountain. I don't know how we survived, but somehow we did. But kids now have these metal or mostly metal water bottles. That's good. Metal or glass, that's preferable to drink from as opposed to plastic. Now, it also depends on timing and temperature. One of the problems with bottled water from plastic bottles is that it's often been in that bottle for a long time, giving it more time to leach out chemicals and plastics, but also heat. If these water bottles have been exposed to heat, and very likely they have in the process of shipping, that also is going to mobilize these chemicals and leach them into the water a little faster. So it is in our best interest to just be a little careful with regards to the lotions, the detergents, the bottles, et cetera, that we're using, just to try to cut down our exposure as much as we can. So in today's lecture, to really wrap it up, we covered some of the fairly complex, hopefully not too complex, ways that microplastics and their associated chemicals like BPA and phthalates can impact fat cell biology and vascular physiology as well. So from adipocyte hypertrophy and chronic inflammation that comes from it, it is clear that these small particles have a big impact on metabolic health, and reducing our exposure to microplastics and the associated chemicals could and should absolutely be a meaningful part of our strategy to improve our metabolic health. All right. Hopefully this information has been helpful. I invite you, if it has been, to share it with a friend. Until next time, more knowledge, better health.